Okay, mine was biology in a box, and this one goes all the way from introductory DNA all the way through very complicated genetics. I chose to start with just the introductory DNA. Okay. Maybe not. Okay. All right. This box is called, it's in your genes, biology in a box. It has and I left the booklet back there. It has a booklet that gives you the whole thing, all the way from the beginning, all the way through very complicated genetics. When I say very complicated genetics, it gets into epigenetics and environmental control and all of that, which is a little bit further than I go in detail with the introductory biology. I do credit biology, but not with introductory. This lesson plan is primarily for first year introductory biology. The topic is on DNA. I just focused on the structure of DNA, replication, and just kind of introduced the function of DNA. Why do we have DNA? What does it do? Learning goals. Under the structure of DNA, I want him to understand that it is a double helix model, which you have here. You can stretch it out and show it. That's kind of neat. I've got one of these I made myself hanging from my ceiling. Very similar to that. I want to talk a little bit and to put numbers into it. I like to tell them that you can take all the DNA in your body, stretch it out end to end, and you've got enough to reach the moon and back 100 times. Kind of blows their mind. You've got almost 2 meters of DNA in each cell, 70 trillion cells. Do the math. We talk a little bit about the fact that we have 6 billion nucleotides in each cell. It goes into detail here. A gene has, I can't remember the exact numbers, gives you the numbers in a gene, the number in an average chromosome. They can use the math to figure out how many genes do you have. Not real, because all your DNA is not genes, but it kind of gives them a chance to play with the numbers. It also, you can take this model and stick it down there by a marble and say you've got to put this DNA in that marble. How are you going to do that? And so it gives you a chance to talk about how DNA twists itself and wraps itself and the histones and all that type of thing. It gives them a little bit of a visual. It's got a set of, I think I left those back there. Oh, no, it's got a puzzle set where you can put DNA together. Now, I have a kit of connecteds that they can make DNA, so I would probably use my kit. It's much more handy to work with. But you can start building nucleotides, and then you can take the nucleotides and show how they stick together to form the double strand, and then, of course, twist it and make the double helix. This one gives you a good opportunity to show the nitrogen basis. It has kits. Now, these are already put together, but they come unput together. And you can pull them apart and let them put them together themselves if you want to. It gives you the example of your purines and your pyrimidines, the 2 rings and the 1 ring. And you can spend a little bit of time showing them how if you put 2 rings together, it makes it pretty wide. If you put 2 single rings together, it makes it pretty narrow. DNA is straight, so you always put a double ring with a single ring. I don't get into the pyrimidines and purines in much depth with my regular biology, but I do introduce that. It gives them an idea of why A goes with T and C goes with G. And then I would start introducing that complementary base pairing at this point, and they would build their double strand matching up A with T and C with G. The next learning goal in this one would be, this is just some pictures that are provided in the biology of the box. DNA replication. I have them build a double strand. I give them a sequence on the board. They build the double strand based on that. And then they take it and break it apart and replicate it. And they can visually see at the end they have 2 strands that are identical, and kind of get that idea how DNA works with complementary base pairing. I would spend a little time discussing the reason for it. well, let me get into the lesson plan. The third learning goal is the function of DNA. Now, that comes a little later. I get into the overview of the function and the central dogma, which is DNA to protein to, or DNA to RNA to protein. It gives you a good opportunity to mention mutations and mistakes that can be made and what results from that. And then the last part of it is inheritance, which would come as a separate unit, really. Okay, lesson overview. And the structure of DNA, I would start with just a general overview of DNA, maybe a little film showing a little bit about DNA. And then we would spend some time manipulating the double helix and seeing how it's put together size-wise, compare it to a nucleus and that sort of thing. And then a little discussion on the total base pairs and do a little bit of rough math on that just to understand the volume of it. Continuing on the structure of DNA, we would build the DNA molecule all the way up to a double strand. I usually use 6 sequences. That's enough to kind of get a feel for it. And then I would spend the last part of the structure of DNA enforcing the purine, pyrimidine. A goes with T, C goes with G. The second topic was DNA replication. We would now take this double strand, break it apart, replicate it, get an idea of how it's done, and then we would spend a little bit of time discussing why it's done, where it's done, when it's done, the fact that they don't just replicate because they get the urge. Well, they do get the urge, but they get a message that gives them the urge. There's a reason for replication. It occurs in the nucleus. Why is it in the nucleus? And that sort of thing. And then the mathematical problem on number of bases showing. I like to give them, just throw this out there, and I'm amazed that some of them get it. I tell them that 30% of the bases are A's. How much is the rest of them? And some of them have no clue. But there'll be those 2 or 3. Now, my honors can usually get it pretty quick, but there'll always be those 1 or 2 that gets it, and they're so proud of themselves. But it shows an understanding of the connection between the bases. And then mistakes. They had a good, and this doesn't require any material, it's that old, it's called by a lot of names, but you divide your class into 2 groups, and you whisper a sequence of 6 bases to the first one, and they whisper it to the next one, and to the next one, and to the next one. And so at the end, you write down what you started with and what you ended with, and you invariably will make some mistakes. It gives you a chance to discuss that mistakes do happen, and that it does have a repair or an editing process, but still some get through, and then you can discuss, oh, what does that mean? They're not going to come up with real good answers now because they don't know anything about protein, they don't know anything about genetics yet, but they can understand that it's important. We can calculate the rate of mutation for each group, so it gives you a chance to use a little math there, and then discuss the result of mutations. As far as assessments go, I like to question a lot. Question as a group or as a whole group when we're talking. Question individual groups when you're walking around. You know, just ask them a question or 2, see if they're understanding what they're doing. Look at their models. Are they making them right? Gives you a good idea whether they understand it. You know, we use exit tickets a lot, just a question at the end of the day, see if they've kind of picked up on it. And, of course, eventually it'll be part of a unit test at the end. I think that's it. All right. I was impressed with this. This goes all the way through the ears of corn, which gets into epigenetics and all of that. So this was pretty complete. It gets into human genetics and all the human traits that you can see, you know, and the effect of the environment on traits and all of that. So this is pretty complete. I'm going to get it this year when I do my genetics unit. Absolutely. till somebody else wants them. Right?
Yeah, exactly.
I guess it would be good if you could give an idea when you get them how long you want them,
Yeah.
so they could
I
plan.
think it depends on your box keeper, you know.
Yeah,
Talk to
in
your-
our place, they're not used a lot, so I've not had a problem getting them and keeping them.
You guys are up.
So we all teach different levels of biology, and so what we're trying to do is create some kind of lesson for CP honors and IB, where we can just do various things with it. And we started looking at we started looking at some data nuggets on invasive species, and then we're trying to incorporate the STEAM with the data nugget, and then kind of got away from that a little bit and focused more on the STEAM part of it, competition between species, and brought the invasive in maybe as a follow-up lesson. So these are pretty much straight from Tennessee standards or the Tennessee lesson objectives that we were looking at. Obviously, interpreting graphs from STEAM is pretty intense. And then we started talking about how we can manipulate or have them manipulate environmental factors to change the relationships between the two populations. Without previous knowledge of population, environmental factors, carrying capacity, and the way different species interact, it would not be of any value to them, because what we found with the STEAM is they've got to have a lot of background knowledge going in to actually use it in a way that would be valuable to their learning. So we talked about, for some of the, maybe your CP kids or go a little over, print some graphs from the program and let them interpret it. And then actually, or even stepping the kids through these. Yeah. So this is the graph between the two species. You want to talk a little about what it is?
So we could, first one thing you do is just give them all this data that we have on our computer, and then give it all and just have them and discuss it. You maybe model what's happening and what they see, what's going on.
first data nugget is new invasive species escape their enemies, and it's really a level 2, so that kind of falls into middle school. But following up with all these graphs, talk about maybe going through the labs, and then how would an invasive species benefit, and then going back to the graphs and saying now you have a better idea of the parameter that you could enter in that would illustrate. Now, not actually do it because we don't have time to eat. You know, we couldn't actually even do it, but we could get him thinking about how to do it. So, that's it.
Okay.
We forgot to mic the last group. Are we going to mic this group?
Oh, I'm sorry.
Oh, that's right.
You didn't get it from me when I left.
Is one of you going to be doing a lot of the talking? Or you can just pass it back and forth. How's that? Who's going to start? All right, just pass it back and forth.
All right, good morning. All three of us were really impressed with the tools we saw on the first day, the Gapminder and the World Mapper. And we really, I think, maybe I'm speaking for them, but struggled a little bit with how we would incorporate this in our classroom because there's just so much info. And I think between us, we teach AP Environmental Science and AP Biology and some regular biology. And time is always that factor. I mean, you can't just turn the kids loose and just say, go figure something out. But what we came up with was trying to use a more guided approach and combine those two resources and maybe keep it just like a 2 or 3 day sort of lesson here. So if you want to chat a little bit.
Well, really, we found a pretty good range of different topics we could use this for. Like in AP Environmental Science, I teach. There's a large unit for population ecology, studying populations and cultures and how they affect each other and other species and things like that. So this could be useful there. Also, like I say, community ecology, agriculture data. You could even transition this into looking at energy pyramids, because as we were playing with Gapminder the first day, we saw that you could look at things like agriculture and disease and those indicators, I think they're called. So you could actually, use this in your, when you talk about ecosystems and food chains, food webs, things like that. And then also when you talk about immunity, disease transmission, maybe you teach A&P or something like that. This could be relevant in that.
Okay, learning goals, objectives. This is there's a number of things you could use this for at the beginning, at the outset, to kind of reinforce that nature of science learning, and like we're talking about, developing questions and hypotheses that the students can generate just from playing around with Gapminder. But they're really conducting research on their own and analyzing the data, making the inferences. One thing that we talked about from BSCS is something called an explanation tool, which we've talked about before, where they're coming up with a question, but they have a piece of paper they have to fill out that they have to make a claim related to that question, and then they have to collect evidence and then have some reasoning at the end as to whether that evidence supports the claim. I think that's a really powerful thing I'm going to do this year that I've not done in the past. But to follow up on that, you can have the students put that on a poster around the room and then have their peers walk around and put little sticky notes on their explanation tool to critique what they've come up with. And it was a really powerful way to learn and then revise those hypotheses, revise their science. So that's something I'll probably do this year a lot.
This came from the World Mapper, and once again, there's just this tremendous amount of data. But I was thinking about my immune system unit in AP Bio, and I thought this was a really neat bit of data down here where they've got the exposure outbreak time has these percentages, and I couldn't make any sense of it, and I still really can't. Why do Russia and in South America, Colombia, have really high proportions of time with influenza outbreaks? I can't think of any real connection between them. But there's some, countries with high values and countries with really low outbreak exposure time. So as far as like a guided inquiry, instead of just turning kids loose with this tool, show them this data and have them start generating the questions. What are some possible reasons you might see countries that are relatively close to each other, Italy with a high rate and Greece with a very low rate? Why should that disparity exist? And let them see if they can't mine the Gapminder and find out some sort of possible explanation for those, and then go ahead and do the poster tool for that. So just one example that just found in kind of a hurry clicking through, but I thought it would be a pretty powerful one to use in class. Yes, sir.
So one of the things you might look at, a woman from Italy has looked at this with the transportation density in those countries.
Sure. I said there's a myriad of factors I think the kids would probably generate and come up with.
She's produced some really nice graphs.
Okay. Yeah, and I'm sure you could also be pointing into Centers for Disease Control and World Health, so there's a lot of opportunity for research just with what would seem to be just one simple screenshot of a question. Yeah, this will probably be blown up a little bit, but that claim evidence reasoning tool, there's lots of rubrics for it out there on the web, and this was just one that pops up on the National Science Teachers Association website. Did the child make a claim that is answering the question? Did they provide some evidence? And what is their reasoning? How strongly does it support their claim? So it's a pretty straightforward tool for assessing, but there's a lot of different variations, and you can certainly tweak them. So that's kind of how we had it plotted out, I suppose.
And Nick, too. Just pass it around as you're talking.
But the man worked to put a sign up when women were, signed. So,
Who's going to talk first? Why don't you start?
Okay, so we decided to look at understanding scientific inquiry, and we used, what was the site? The data nuggets and the Understanding Science 101.
This was one of the links that was off of DataNet.
Well, at my school, we have to have warm-ups and we submit those weekly. So I just went ahead and put up, on the Data Nugget lesson plan, it said to have them just write a definition of science. Well, if you do this within the first 3 or 4 days of school, you're not going to get very good quality. So what we usually do is actually give them a definition and have them explain what they think that definition means. So you don't have to use, of course, the ones that I did for the warm-up, but that's an option if you want to. And then we put both learning goals up there, a state one, which I think was from Tennessee. And then, well, we've already adopted the Next Generation Science Standards in Kentucky, so we went ahead and put up the practice numbers that it goes with, because it's not a core content number, but it is the scientific practice numbers. So we went ahead and added the 3 that it falls under for that. And then what what my students do is go ahead and create flashcards, and we get the little binder ring clips, and we have hole punches, and they create flashcards that they can then just flip through when they study. So once again, that's an extra part. This is the overall complete lesson link, so the link is in there for the lesson. And then they had a game called Checks, and this is just basically step-by-step for the kids what to do and about how long they should have for each. The link right there is the link to the lab and the PDF for the checks to print out. You can make your own, but that's the link if you want it. These are the articles that they had to go through. What was it on? It was on
the meteor or the asteroids
and dinosaurs. So they go through the article and they highlight wherever they think science is going on. Okay. Short version was about 1.5 pages. The long version was 8 pages, and it included graphs and pictures and all that. So if you have a 50-minute period, which is what I'm going to this year, the short version is going to happen. Last year I could have used the long version, but not anymore. And then we were thinking about handing out the PDF, which is linked to it, but I went ahead and put it up there as well. But they'll have a handout. A couple options is you could just give them the paper handout, but you could put it in protective sheets so they can have a dry erase marker and mark where they're flowing through it, or you can laminate them. And once again, they can draw arrows of where they are following the science through the article, because the next step is to actually go through and find where they were doing science and trace it through the scientific method. And then there are 2 formative assessment options, an exit slip where they go back to their original definition and talk about it again, and then you have a homework where you can give 2 versions, a short version of DNA and then the long version, and they do the exact same thing that they did in class, but for homework.
I wanted to add, HHMI has short film on both of those resources, the double helix, which was the discovery of the double helix and the scientific process that this Watson and Crick went through. And they also have one called The Day the Mesozoic Died, which is all about the asteroid impact and the evidence that scientists have that that's what killed the dinosaurs.
There's also a click and learn on
Yeah.
science too.
Yeah. So there's a lot of resources on HHMI for those 2 topics in particular if you wanted to get more detailed.
This whole topic, understanding science, of course, goes to all types of other classes as well. So I'm planning on using this basic outline and everything, especially for the KT extinction in my geology class. And since I'm teaching biology and geology, I'm trying to find links between the 2, and this works great.
How about this one?
And if you'll just pass it around as you talk. Thank you.
Okay. What happened? I don't know. Okay. We had considerable help from John as to how we could extend this for different level courses. This is obviously applicable to any level of biology and in environmental science, of course. I assume everybody who teaches bio does environmental science, at least to some extent. So biology, ecology, biodiversity, how does energy flow through? These are some of the goals. How does energy, what we want the students to understand, how does energy flow through a diverse ecosystem? How do the relationships between different organisms affect the flow of energy? Let's see, lesson overview. We went back to the three of us talked, and we obviously wanted to use as much as we could from what we learned here, and we all like the idea of opening with a scientific method kind of approach. What do you already know? You know what your not problem is, but what you're dealing with, the question or problem or issue. So what do you already know? But the same way we've been doing in here. What things are you already aware of, or can you figure out very easily without doing an experiment? And then, of course, now what do you need to know? What things do you need to know? And you wanted to go through the Mike.
So a couple of things with food web. So we want to work through a couple of different examples as we go through. And the first one is a modeling exercise that John mentioned, but I actually do in my class, where I give all my students different organisms and a bunch of yarn, and they have to build a food web out of all the connections that they build, which is a very interesting process. And one of my students last semester made the comment, "This is the biggest CF I've ever seen in my life." And because one of the things I think my goal in ecology is to demonstrate how complex it actually is. A lot of people think ecology, oh, trees, rivers, fish, deer, yay. It's actually fairly complex. So that's a great modeling exercise to kind of get them used to the idea of the complexity of a food web. And then we've got a simple model that we'll expand on here. So we just picked a very simple food web that touches through a few organisms as we go through. And what we then create was a matrix based on predator-prey. And this is where John's insight came in. And so we have predators on the top and prey on the bottom, and so we can actually build that. And so we'd walk this through with our students as they kind of built this. And then that would, as we set that up and scaffold them, that'd go on to the next part where we provide them a more complex food web. And this is where we started as we did this, so then we built this thing. Yeah, and that's the one John gave us, so we can blame John for that. So we basically did the same sort of thing with that. We took the food web and we looked at predator-prey, and so we built our matrix yet again, which is a little more complicated. And the way the matrix works is if a predator eats a prey, you put a 1. If they have no interaction, you put a 0 or leave it blank as we go through, so you can start seeing the interaction as we go through. So our assessment, after we've kind of modeled and we've worked our way through and scaffolded this, assign a group to perform the same analysis of a food web in a biome of their choice. By high school, I don't really focus a whole lot on biomes. I figured if you don't know biomes by now, then you have serious issues. Although I do work them in through ecology, so I think by isolating the biomes and then looking at a food web as they go through, and they can create and present their results just like we did in class. So we kind of worked through in groups and did this, and it can be graded on a rubric. And then there's an extension that maybe John can give a little more insight to after this, because we try to follow. In the STEAM thing, there's a Java Benzer process where you can look at overlap and niches and rules as you basically put the interaction together. You click these boxes, and then you can drag them and isolate them to see more about the patterns, I think. And John, I don't know if you want to elaborate on this a little more.
So what is a niche? And in a community ecology, if two predators eat some prey in common, that becomes part of your community niche. And so what we're trying to relate are niches and trophic levels and energy and biomass kind of thing in these organizations.
Yeah. So we took our matrix, put it into this, and we can analyze it and look at our patterns of squares where we can see where there's commonality of what.
There's documentation on the Esteem website.
Voila.
Okay, so we, as it says, built on phylogenetic trees, and with those phylogenetic trees, this particular set of lessons is designed more for an advanced or an AP type course. And so, sure. The learning objectives, sort of taking kids through, they're going to learn how to do gel electrophoresis and how to create a sequence. They're going to learn how to analyze the sequence to make matrices. They're going to learn how to take the matrix and turn it into a phylogenetic tree. They're going to learn how to use the national databases and online tools to interpret that data. And ultimately, we're leading to where they develop their own research questions and use these tools to answer their own research questions. So we tried to build this in a sequence, if you would. Go ahead. So on day one, we would do an introductory activity, which many of you experienced yesterday with the great clad race, which is something that I've already used a number of times. Kids really get, I feel, a pretty good grasp of the basics of a tree using that activity. I ironically had sort of made a small modification by bringing in real animals. I'm glad to see the update. I like theirs better than mine. So everybody already went through that, so I won't explain that. And then the 3D model with the pipe cleaners and visualizing that all sort of lays a foundation for, okay, what is a tree? How does a tree get sort of put together? Those types of things. Then we would go to the basic math behind the tree, which is taking off on what Tony talked about yesterday. I liked how he will sort of walk through the sequence. It was very logical, very sequential. Kids sometimes don't get an idea. I know when I originally did this and was using the more complex programs, kids didn't get where this was all connecting. So we would analyze a given DNA or amino acid sequence, just like we did yesterday, determine the number of sequences, create a matrix C, from that matrix C create a tree, using EVOS evolution sequence. We could bring up different types of sequences and do those types of things. So this was pretty much what we did in class yesterday. Probably follow that up with a few other scenarios they could take home that evening as practice. Come back, present their stuff to each other in small groups, debate, talk, those types of things. Part 3 is something that I've been using. It's not original. Biorad, if you're familiar with Biorad, Biorad puts out a kit called the Comparative Proteomics Kit. They used to call it Fish Proteomics. And I like this one because it goes all the way back. So you take just small pieces of fish. In this case, we're comparing the, I believe it's the myosin protein in the muscle, which is highly conserved. And so you extract the myosin, you put that into a protein gel, you run that, they can see the bands, they analyze the different bands for each fish, then they figure out how many bands each fish has in common, and now we're back to create the matrices, create your own tree. And I like that because they're going all the way back to where the original data comes from, and they can sort of walk through, okay, first I've got to figure out the sequence, then I've got to analyze the sequence, then I've got to build the matrix, and then I've got to do the alignment, and then I've got to, so it does a nice job of seeing how to create this. So from there, next, that's all right. So from there, then we introduce them to, you don't always have to create these from scratch. There's huge national databases out there. There's probably somebody that's already done a lot of this for you. And we introduce them to how to find gene sequences and then how to put those gene sequences into an analyzing tool. Lately, I've just been simply using the NCBI site to get the sequences and been using phylogeny FR. It's much simpler than using molecular workbench for the kids, and you can get pretty quick trees. So once we've introduced them to these tools, I would not do all of these. These are some samples. Okay. There's an old one from Kim Fogel's site called Whales, Walruses, and Seals, basically trying to determine was there a single evolutionary event that had aquatic mammals evolved from land mammals, or was it a series of convergent evolution events that took place? There's one that Bob does.
Yeah, the polar bear activity is one from ABT. And the kids have to ask two questions, or ask two questions. Is the polar bear derived from the grizzly bear? And are giant pandas true bears, or are they related to raccoons? And the original activity took them through biology workbench to answer those questions. And I modified it this morning to use phylogeny FR, which is much more efficient for the kids. But before I have them do that, I would have them work in EvoSeek so they have some idea of what's going on, because it's black box. It's like magic. The tree just pops up. Then in terms of assessment, I use, when I do that activity with them, I use the explanation tool that was mentioned earlier.
And these are just some other examples. There's an old one out there. I love Calvin and Hobbes. There's a strip where he went and did this made-up book report about bats or bugs, and sort of embarrasses himself, gets in trouble. The scenario is, okay, the teacher gave him a second chance. He's supposed to redo the report. Now he's claiming that bats are birds. Is he on the right track? Is he going to get away with it? Kids sort of like that angle. We can do a primate analysis. We can do the old reptiles and birds analysis. There's lots of ones you can do as an example. I like to do stuff, especially in the AP, where we're going to return to it later in the year, not just do one and done. So with the HHMI lizard, there is a phylogeny that's done with the lizard activity, and that I'll actually come back to maybe 2 or 3 months later, and we're repeating and coming back and exploring and connecting an idea from the past, which is something that I'm a big fan of. Assessment-wise, students could eventually develop their own research questions using the national databases and tree building programs. They would answer their research question, then create some type of mini poster presentation, slide presentation, where they have to present it to the class, defend their answer, provide the evidence for the answer. There's lots of different ways you can do that. Go the old testing situation. These are just a couple different ways you could put the test on the question, where they're having to interpret the data, not just a simple multiple choice type thing. I think that was it. So,
I spent some time thinking about sequencing this morning. Normally I've done the Great Clade Race, the older version. I'm also going to upgrade to the newer version. But I've done it in the phylogeny section much later in the course, and I think I'm going to move that up right to the beginning of the course. I usually teach, I spend more time in Chapter 1 than most people do, and I give them a sense of some broad themes, and then they just keep cropping up over and over again. And I think I'm going to move that up so they have some concept of trees, because it really, I think as Drew said, it takes away the biology and just gets at the essence of what tree building is about, and then gives them that sense. So then I'll just refer to that all the way through the course. So I guess that's it.